Welcome to the last segment of this bioinorganic chemistry module. So now we learned that oxygen binding is a very crucial thing in the bioinorganic chemistry, and we learned the basic chemistry behind this oxygen binding through the hemoglobin. Now we will take some interesting examples of this oxygen binding to hemoglobin and how it affects our daily life. So we will take 3 questions. The first question, over here I am showing the oxygen binding curve for the myoglobin in the dotted line, which is as expected a hyperbolic interaction. Then I am showing you hemoglobin binding to oxygen at 3 different pHs, 7.6 in the blue, pH 7 in the purple, and 6.8 in the orange. And as you are seeing, as I increase the acidity or decrease the pH of the solution, the oxygen binding at lower pressure for hemoglobin is getting down. Question is why? To answer that, we have to again look back to this interaction between the different subunits. So as I already told you earlier, that the different subunits of hemoglobin have a very nice interaction between them, and that is mostly coming from hydrogen bonding and ionic interaction. Now over here, which of the amino acid mostly take part through this inter-subunit interaction? A key player to that is histidine, and histidine has a pKa of around 7.5. In histidine we have imidazolium group, and in the acid base chemistry you come to know that imidazole can be a very nice system which can act as a very strong acid and base. And this imidazole connected histidine gets protonated once you go below 7.5, and as it gets protonated, it now can create more hydrogen bonding network because it is protonated, you have more protons, and at the same time it is charged imidazolium ion. In the charged form, it can create much better salt bridges, much stronger salt bridges. So what will happen, as I am increasing the acidity or decreasing the pH, I am actually protonating the histidine, and as the histidines are getting protonated, that is creating much more stronger bonding between the inter subunits, and due to this the oxygen binding will have much harder time to break this inter subunit interaction, and that is why as I am increasing the pH of the solution, the oxygen binding needs more oxygen so that I can break down this inter subunit interaction. So that is why we see that particular effect, and that particular effect known in chemistry is the Bohr effect. And what is a direct application of that? So generally when we have starvation of oxygen inside the cells, we produces carbon dioxide as a side product, and that carbon dioxide gets accumulated over there. And carbon dioxide in cellular system, that means in aqueous cells, produced carbonic acid. Carbonic acid is a very strong acid to change the pH below 7, and that actually makes sure that this oxygen binding to hemoglobin is weaker. And how that actually helps? That actually helps, if you look closely over here, you can see at cellular concentration the hemoglobin is not like devoid of any oxygen, it has some oxygen present like 20%. But if I increase the acidity of the solution, the minimum oxygen binding of the hemoglobin gets down. That means it cannot hold the oxygen very well. That means whatever oxygen present in the hemoglobin in the cell, it will be bound to release that oxygen in the cell. So that means cell is trying to use whatever oxygen available with the hemoglobin there and use it. And that is actually a self defense mechanism. When we actually devoid of oxygen, we try to make sure to get out most of the oxygen present with this oxyhemoglobin. Now come to the second question. It is found that hemoglobin actually transfers more oxygen if we go to the higher altitude or we do a lot of exercise. Why? The answer of this question is hidden with this molecule shown in the left hand side, known as 2,3-biphosphoglyceric acid or 2,3-BPG in abbreviation. So whenever we are in stress, we are doing a lot of exercise, we go to very higher altitude, our cellular system itself releases this particular molecule called 2,3-BPG, and this 2,3-BPG, as you can see, it has these phosphonate groups and acidic groups, carboxylate acid groups. It actually binds at the middle of this hemoglobin structure. And what is its effect? Once it binds there, the phosphonate groups, the carboxylate groups, they started forming ionic interaction with the amino acids present over there, that means on the surface of the different subunits. And this 2,3-BPG act as a glue to make sure the subunit interactions are getting stronger and they are all closely packed together. Again, it has the similar effect that once the 2,3-BPG get released in the system, it makes sure that the movement of the hemoglobin from a very tightly packed state to a relaxed state getting more energy demanding. That means it needs more oxygen. On the other hand side, we can explain that whatever oxygen present in the hemoglobin, it will get released. So again, over here you can see in presence of 2,3-BPG, the bright blue color, it actually releases more oxygen from the hemoglobin in the cellular system. That means it is actually make sure to release all the oxygen present in the hemoglobin. It's not allowing it to keep a lot of oxygen, and it makes sure that our body gets enough oxygen for survival in the high altitude or during the high exercising under stress conditions. Third question, it is found that if I have a free heme, that means only a iron porphyrin system, it binds carbon monoxide way too strongly than oxygen. It binds oxygen almost 25,000 times stronger. But the same heme, when it is present in hemoglobin, it binds oxygen still weakly compared to carbon monoxide, but it is only 200 times. So it is almost more than 100 times change in the binding affinity when the hemoglobin is present. That means the protein structure is present. Question is why? The answer over here lies how the hemoglobin actually binds to the oxygen. So far you can see all the structure we have shown, the oxygen, a linear molecule, doesn't bind in a linear fashion to the iron. That means you cannot put iron and the two oxygen in the same line. But on the contrary, it actually binds in a bent fashion. You can see there is an angle between this iron-oxygen dihedral plane and this oxygen-oxygen dihedral plane. So why this bent bending? This bent bending of oxygen is actually helpful, because as you learn from the molecular orbital structure of oxygen, oxygen has two unpaired electrons, both in the pi star orbital. One of them act as the interaction through this sigma interaction, one of them in the pi interaction. During the sigma interaction, one of the pi star orbitals shown over here will interact with a vacant d orbital of the iron center in the hemoglobin, the dz square. And if it binds in this bent form, then the orbital interaction is better, the orbital overlap is the highest. If the oxygen was linear, then there wouldn't be this kind of direct overlap. And this overlap actually gets supported when you have this bent interaction. That is reason number one. Second reason is the interaction with the pi star orbital. Again, the pi star orbital is a similar pi star orbital like this, but in the perpendicular plane, and that also interacts favorably with good overlap with the iron dxz orbital. They also have very good pi interaction with good overlap. And both this interaction happens favorably in this bent form. And the other way, obviously, this histidine over here interacts through the hydrogen bonding and makes sure in the bent form they have enough distance to create a good hydrogen bonding, because the histidine comes in the way if it binds in a linear fashion. So these three reasons is actually the main reason why the hemoglobin bind oxygen in the bent mode. Now what happens for carbon monoxide? Because of the presence of this histidine again in the binding position, it actually differs the carbon monoxide to bind in a linear fashion. Carbon monoxide would like to bind in linear fashion, but there will be a steric hindrance between this histidine and carbon monoxide if it is there. So carbon monoxide has to bend out, and during this bent out, it also forms a bent interaction with the iron of the hemoglobin. And what is the effect? In carbon monoxide, the HOMO, where the electrons are there to form a sigma bond with the metal D, it is a linear orbital like that. And that orbital interaction is weaker if it is binding through this bent form. It would be much better if it is a linear form like this. However, the presence of this distal histidine makes sure that the carbon monoxide cannot bind straight and it has to bend its way out. It actually lowers down the overall overlap between the iron and carbon monoxide carbon. Whereas on the pi star orbital is doing a very good pi interaction is fine, but the sigma interaction goes down when it goes to this bent form. And due to this presence of distal histidine and due to the unfavorable orbital overlap, carbon monoxide binding gets weaker, what it's supposed to do in normal linear condition. So that is the main reason. The binding spot assures that you have to bind in a bent position rather than a linear position, and that is the main reason why carbon monoxide binding gets weaker. And what is the effect? The carbon monoxide binding to hemoglobin is an irreversible process. Once it binds over there, it binds so strongly it doesn't come out. So if we have carbon monoxide in the atmosphere and if we inhale that, we will die because it will take care of all the available hemoglobin. So we cannot transfer oxygen from the lungs to the cells. However, due to this bent binding mode, it actually weakens it. Otherwise, what would happen? We would die even with ppm level of carbon monoxide. But now, due to this weaker bent bonding, we can still survive up to 20 to 30% of carbon monoxide. The lethal dose only is above 60%, which is supposed to be at least 100 times lower than that. So that's why the interaction between the distal histidine and binding between the iron and carbon monoxide or oxygen actually plays a key role to make sure how this carbon monoxide toxicity goes down. So over here we will end our fourth and final segment. I hope now you have a very good idea why bioinorganic chemistry is important, how bioinorganic chemistry actually makes sure that we take the oxygenic course of evolution. Oxygen starts off a poison, but then bioinorganic chemistry itself help us to make oxygen our lifeline, and at the same time it gave us a very controlled way to transport oxygen from the air to lungs and lungs to the cells, and also make sure we actually have enough oxygen present whenever we need through this very unique interaction between the biology and inorganic chemistry. Thank you.